Good morning, distinguished guests, ladies and gentlemen. It is my great honor to welcome you all to this highly anticipated awareness seminar. Before we begin with the introductions of our special guests of honor, allow me to provide a brief overview of the initiative that brings us together today, INSTAL Africa. INSTAL Africa, the Innovation Science and Technology for Action Learning in Africa, is the result of collaborative efforts initiated in October 2023. This initiative leverages the Drosophila melanogaster fly model and state-of-the-art infrastructure to enhance fundamental research capacity in Rwanda. Rwanda has made remarkable strides in advancing its scientific capabilities, recognizing the pivotal role of research in sustainable development and public health. INSTAL Africa was born from a collaboration between the Foresight Institute of Research and Translation, Rwanda, FIRAT Rwanda, and its technical partner, DROS Africa UK, in conjunction with key national institutions such as the National Council for Science and Technology, Rwanda Biomedical Center, the National Industrial Research and Development Agency, the University of Rwanda, and the Rwanda Food and Drug Authority. This collaboration embarks on a 5-year comprehensive capacity development plan aimed at cultivating the next generation of scientists and fostering research excellence across medical, applied, and fundamental sciences. From healthcare innovation to agricultural production, this partnership envisions driving progress in public health and sustainable development across Rwanda and beyond. It is against this background that we convene today, united in our shared commitment to advancing science, technology, and public health for the benefit of our communities. Next, we will move on to narrating the profile of our invited guests.
Ladies and gentlemen, allow me to introduce Dr. Abdul Basit Amin, a distinguished physiologist whose work bridges the worlds of discovery science and translational medicine. For over a decade, Dr. Amin has been at the forefront of teaching, mentorship, and research, training the next generation of physicians, pharmacists, nurses, and life scientists, while advancing new knowledge in physiology, nutrition, and cell biology. At the heart of his research lies a commitment to uncovering the molecular basis of some of today's most pressing health challenges: neurodegenerative diseases, energy metabolism, and host-parasite interactions. Using advanced animal models, his pioneering studies have broken new ground. Among his remarkable contributions, he has demonstrated how the central vitamin D3 receptor can improve motor symptoms in Parkinson's disease, discovered semaphorin 4B, a novel adipokine with major implications for obesity and metabolic syndrome, and explored how host pathogen interactions can be leveraged to develop therapies for non-communicable diseases. His work with plant-based therapies and oxidative stress regulation has further broadened the search for innovative treatments. With more than 50 peer-reviewed publications, over 700 citations, and a strong record of competitive research grants, Dr. Amin's scholarship speaks for itself. Yet his impact goes beyond the laboratory. He has been instrumental in advancing African science through international collaborations, capacity-building workshops, and symposia that connect researchers across continents. From the next Einstein Forum's African Science Week to student mentorship in Nigeria and Portugal, his leadership continues to empower young scientists and strengthen research communities. Currently serving as a senior lecturer in human physiology at the University of Ilorin And as a postdoctoral fellow at the Gulbenkian Institute for Molecular Medicine, Dr. Amin remains deeply committed to bridging basic discovery with translational applications. His innovative use of animal models of neurodegeneration, obesity, and infection continues to open new directions for therapies against some of the world's most challenging diseases. Please join me in welcoming a scholar, mentor, and global voice in physiology, Dr. Abdul Basit Amin. The floor is yours.
So, okay. Good morning. Thank you so much for the kind introduction. So, it was, I don't think I deserve that much of an introduction. But my name, like it was mentioned, is Abdul Basit Amin. So, I'm currently a postdoctoral fellow at the Gulbenkian Institute for Molecular Medicine in Lisbon. And I also teach in Nigeria as a senior lecturer at the University of Ilorin. So the title of the talk for today is the mechanism of blood glucose regulation during trypanosomiasis. Basically, I'm going to explain how the blood glucose is regulated, and then the bigger picture about what we can learn from the interaction between these parasites and the host. So first, Portugal. It's this is me marketing Portugal to you, and I think the most popular person or the biggest export from Portugal is Cristiano Ronaldo, right? But then in Africa also we have some Portuguese influence because at the moment we have at least 5 countries that are Portuguese speaking that are together called the PALOP countries. And for some people, they know Portugal for tourism, the food, the nice beaches. And even in Nigeria, we still have some Portuguese influence. Like Lagos is a Portuguese word for language, so they have some influence all over. But in terms of science, so what do we know about Portugal? I don't think many people know so much about Portugal in terms of science. But a long time ago, not so long ago, in 1949, a Portuguese scientist known as Muniz got a Nobel Prize in physiology and medicine, in medicine and physiology. But then we also have some amazing institutes like the Champalimaud Center for the Unknown, for instance, and they're one of the best neuroscience institute in Europe and one of the best cancer clinics in the world. We also have an institute called the Gulbenkian Institute of Science. So this is just to tell you little about some of the science that we do here. But talking about me now as a person, how I ended up in Portugal. So my education, I started from Nigeria in Kaduna, where I went to the Federal Government College, Kaduna. That's my high school. From there, I proceeded to the University of Ilorin to study physiology, where I did my bachelor's and master's degrees, and then came over to Portugal, came to the Nova University for my PhD, and I did my research at the Gulbenkian Institute Gulbenkian Institute of Science in Lisbon. So I did my PhD there. So, in terms of my research interest, this is where I will take it slow because I know that we have a diverse audience. So I'll try to make it as simple as possible. And then if you have specific questions at the end where I need to expand on things, then I will do that. I do research in neuroscience, which was more before my PhD. In my PhD, it was more focused on metabolism. And then for the postdoc, I saw an opportunity in parasitology. So the disclaimer is that I'm not a parasitologist, but I'm trying to leverage some of my knowledge of physiology to see how we can tackle problems in parasitology, and then the other way on how we can use the things we learn in parasitology to tackle some human problems. So these are the research that I do. So in moving closer to what I will talk about in this presentation, this is more of interest to the parasitologists maybe, but we know that in infectious diseases in general, even in malaria, in trypanosomiasis, which we call sleeping sickness, the target is always to kill the pathogen. You know, we have anti-malarial drugs, we have antibacterial drugs, for instance, like antibiotics. You know, the goal is to always kill the pathogen. This is why we have it here saying that the first strategy is to eliminate the pathogen. But what we also know that we actually learned from plants is that, you know, even in malaria, for instance, the symptomatic stage of malaria is when the parasite gets into your red blood cells. So it's the symptoms that tells you that you have pathogens in you. But then you can also have pathogens in you and actually not have symptoms and don't even realize that you have the pathogens. What this means is that your body is not clearing the pathogens, but your body is tolerating their presence. So we have two options. It's either you clear off the pathogen or the pathogens are there, but you're still healthy enough and you try to tolerate the presence of the pathogen. That's the second strategy. So this will be important in later parts of the talk. But then in terms of the things we try to do now, which is like the innovation of fusing physiology and parasitology, which is more of like the host pathogen interaction. Here we want to understand how the host interact with the pathogen. It's a huge field at the moment. But our goal is to leverage the knowledge we learn from this, not to necessarily tackle infectious disease, which is also important, but to leverage knowledge in other non-communicable diseases like obesity, diabetes, neurodegenerative diseases, and other forms of diseases. You know, it's to leverage this. This is still in its very early stage. We don't have any therapy for this approach yet. So we're hoping to be the pioneers of this concept and the field. So this is all the background, right? So we take a rest a little bit, and then I'm going to tell you the research that I do. So all the data I'll be showing you today will be data that are unpublished. So what that means is, you know, you can ask questions to criticize, you can ask questions to make it better, you can ask questions to ask for clarity on whatever I talk about. But the plan is to keep it very simple so that even if we have a lawyer in the house listening, they take something away from this. So my pathogen of interest is the Trypanosoma brucei. And this Trypanosoma brucei is transmitted to humans and cattle by the vector. That's the insect vector called tsetsefly. We all know tsetsefly, we know trypanosomiasis, the sleeping sickness. So as a child, I always assumed that when you have trypanosomiasis, you will just be sleeping. You know, I never had it, so I didn't know this. But science has shown now that the trypanosomiasis itself can induce a change in our circadian rhythm. So it really has something to do with our sleep. But in what I care about, which is more the metabolic part of the disease itself. So what has been reported in the past is that maybe humans or animals that get bitten by tsetsefly and then they're infected by the parasite often have hypoglycemia. It's different types of hypoglycemia. Sometimes it can be transient. Sometimes it can be the spurious type of hypoglycemia, whereby even if you provide glucose, the blood glucose still remains low. But then, because we use experimental models, I will be showing you the effort we've made to understand this in mice. So basically here you have mice. Here we call them, when I say mice, it means it's just a normal mouse, a wild type mouse. So here we have mice in blue that are not infected, and then we have mice in red that are infected. And it is obvious for you here to see that the mice that are in red have lower blood glucose level, and this blood glucose level is in fed state, so they're not fasted, it's still in fed state. And then because the mice in red are sick, they exhibit a behavior that we call the anorexia associated with infection. It means that at some point during the infection, this is their food intake, they decrease the amount of food they eat. And even in humans, when you eat less or you fast, your blood glucose level goes down. So what that means is that what we see in the reduction in the blood glucose level in the animals that are infected may be because they don't eat as much. But then if you look at them again here, they recover their food intake over time. But independent of this recovery of their food intake, the blood glucose still remains low. And one of the things we want to understand is the why. Because these are simple questions that physiology in a normal context had deciphered. But in the context of infection, we still really don't know what happened. So, and this is what we want to understand. So, but we have two hypotheses. If you have low blood glucose level in an animal that is infected, it may just be because the parasite itself, I'll keep using parasite so that I won't be using the jargons, right? But when I say parasite here, I mean the T-Bucia, because T-Bucia also feeds on glucose. It may just be because the T-Bucia feeds on the glucose from the host. This is why you have low blood glucose in the host. It may also be that the host is adapting to the presence of the parasite, and in some way it lowers its blood glucose, or it can be a combination of both. But because we're talking more about the host and we know a little bit more about the pathogen, we want to know what the host does. So most of the things I will show you now will be about the host itself. So if we assume the host is doing it, but this is just to go back to our basic biology of how glucose is regulated in normal condition. Let's say humans, for instance. Let's say you just had your bowl of carbohydrate, for instance. The carbohydrate is digested. The final product of the digestion of carbohydrate will be glucose. The glucose is absorbed into your blood. Your blood glucose level goes up, right? So you want to normalize your blood glucose level. What that means is that you will store your glucose in different organs. And one of the major organs is the liver, for instance, just to bring the blood glucose level down. So when you're not eating, which is your fasting state, you have glucose that was stored in the liver previously. So you will then mobilize this store to produce glucose that goes into your blood. Basically, what I'm saying is here is this. You have two states, which is either when you finish eating or the point when you're fasting. And then I've mentioned how blood glucose level is regulated. This is a simplistic overview of what's happening, just to tell you that the liver is a central organ to this. So in the context of the infection, what we want to understand is if any arm is impaired, the first arm is okay. In this case, the mouse eats. What happens to the glucose? Does the glucose go into the blood normally or not? So the simple experiment you will do, there may be more complex things, but like I mentioned, we keep it simple. The simple experiment you can do is to give something that needed to be broken down into glucose to the animal. So what we do here is, again, the normal animals that are not infected are the ones in blue, the ones that are infected are the ones in red. So you give them sucrose that has to be broken down into glucose, and then you look at the level of glucose in the blood. What you expect is that you give sucrose, it's broken down, the glucose level should go up, and over time, it is now cleared. So what we show here is the percentage change in the blood glucose from the basal level of the glucose. And what you can see here is that there's no difference between the infected animal or uninfected animal. So we decided to go to the next level, which is that when you have the glucose in the blood, do the tissues that take up glucose really take it up, or there is a defect when an animal is infected? So in this situation, what you do is you give glucose to the animals after being fasted for a few hours, like 6 hours, and then you see how the glucose is cleared in the blood. Basically, you give glucose, the glucose in the blood goes up, and then it is cleared over time. Again, you will see that whether the animal is infected or not, this is not affected. There's no difference at all. So the last arm that we then looked at is the ability of the animal to make glucose. So the ability of the animal to make glucose, one of those ways is called gluconeogenesis. And basically what you do is like you have some substrates that are not derived from carbohydrates like pyruvate, glycerol, and amino acids that the liver can use to produce glucose. So a simple experiment again will be you fast the animal, and then you give one of the substrates, although we give several substrates, but I'm showing you one here. And that substrate, for instance, is pyruvate. And what you expect is that as soon as you give pyruvate, the liver should start producing glucose. And so the blood glucose level will go up and then get cleared over time also. So what you can see here is that the blue animals, which are the animals that are uninfected, the normal animals, are able to produce more glucose relative to the animals that are infected. So the summary here is that it's possible that what is happening here is that the animals that are infected are deficient in producing glucose. And this may contribute to the hypoglycemia that we see during infection. So to really understand this, I mentioned that you need substrate to make gluconeogenesis. So in the normal animals that you give nothing, and then you infect, if you look at the level of some of the substrate that the animal can use for gluconeogenesis, just a simple one like pyruvate and glycerol, you will see that there's no difference at all. So this is an uninfected animal, and these are animals that are infected on different days post-infection for you to see how the level progresses as the infection progresses. There's no statistically significant difference in the level of pyruvate, and the same thing for the glycerol. So what is causing the reduced gluconeogenesis in these animals is definitely not because they don't have enough substrate to make the gluconeogenesis. So we looked at the level of different hormones, but the one that is most common to people is like insulin, because we know about diabetes. So what insulin does is that it decreases gluconeogenesis, and what glucagon does is that it promotes gluconeogenesis. So if you compare the level of uninfected animal, the level of the insulin in the uninfected animal and the infected animals at different days post-infection, there's no difference. But when you, and I said insulin is the one that negatively regulates gluconeogenesis. So the positive regulator of gluconeogenesis is glucagon. And what you will see is that if you compare to the uninfected animal, the infected animals on different days post-infection, you have this increase as infection progresses. So this shows that the animal is really trying to increase gluconeogenesis by increasing the secretion of glucagon itself. But systemically, this is not happening. So there must be a repression somewhere that overrides the action of glucagon itself. So because the liver is the main organ we care about now, we want to look at the liver itself to understand how responsive or irresponsive it is to the hormones and the substrate for gluconeogenesis. So this is to make it very simple. I can explain better if anyone wants to understand it better. But what we did here was to take the whole liver of mice, get RNA from the whole liver, and do an RNA sequencing. So basically what we're looking at here is that we're looking at the expression of all the genes in the liver of these animals at the level of the messenger RNA. So I hope that is, so at the level of the messenger RNA, so the biology people will understand this. So it's to look at what's happening in the liver as a whole. So, and this is a principal component analysis kind. So to make it simple, what this is saying is anything that is in a circle are similar and things that are in another circle are different. So, but the relationship is this: when you have the horizontal difference between the different circles shows that there's a huge difference between them. But if you're on the same vertical line, it means you have less difference between you. So, and what you can really see here is that the uninfected and non-infected animals are clearly different from, which are in green, are clearly different from the orange, if I get the color right, yeah, orange, and the blue. So the blue are animals that were infected for just 6 days. The orange are the animals that are infected for 10 days and 22 days. So you will see that the liver from the animals that are infected for 10 to 22 days are quite similar. They're a bit different from the ones in blue, but they are quite similar. But then the ones in green are completely different from the ones that are infected. So when you look at the genes in this liver, most of the genes that go up, which we say are upregulated, are related to inflammation, which is immune response, right? And the genes that are downregulated are mostly related to metabolism. And because we've been talking about gluconeogenesis so far, so I'm going to focus on gluconeogenesis itself, which is a metabolic pathway on its own. So we wanted to understand what happened. So to make you understand what happens in gluconeogenesis a bit, I already mentioned that you have different substrate for gluconeogenesis. So you just have this pathway that converts pyruvate or lactate to glucose. And then you have another pathway that converts amino acid and glycerol to glucose. But irrespective of where the substrate is coming from, the last enzyme in the step of conversion to glucose, which converts glucose 6-phosphate to glucose, is called the glucose 6-phosphatase. Without bothering with other genes, so just looking at this gene, We've seen this in RNA-seq. We want to confirm this. So you just do a real-time qPCR, a real-time quantitative PCR to quantify specific genes. So with the RNA-seq, you see all the genes. You're checking all the genes. With the RT-qPCR, we're selective of the genes we want to look at. And then the gene we're looking at is involved in gluconeogenesis. It's like the master gene for gluconeogenesis. And what you will see is that in an uninfected animal, which is orange, so what you're seeing here is the fold change, whether it's going up or down, basically. So you will see that as the infection progresses, the expression of this gene goes down, meaning that with the progression in disease, which we see that we have a repression in gluconeogenesis, you also have a repression in the expression of a gene that is important for gluconeogenesis. Basically, what we're saying so far is that the gluconeogenesis that we see repressed may be organ-centric, since we see all this in the organs. So the simple thing you want to understand now is, what is the driver for this repression in gluconeogenesis? So I mentioned earlier that you have this upregulation in immune-associated genes. But because we see the repression in gluconeogenesis in later stages of infection, and we know that the immune cells that are more important for those stages are immune cells of the adaptive immune response. Basically, you have two immune responses. You have the innate, you have the adaptive. The innate is the immune response that is triggered as soon as the pathogen gets into the host. But the adaptive immune response, which is more a function of the T cells, these are the T lymphocytes, and the B lymphocytes make up the adaptive immune response. So the other hypothesis will be that maybe the parasites themselves are the ones repressing gluconeogenesis. So we have two hypotheses now, that the adaptive immune response represses gluconeogenesis, or the parasites themselves have the capacity to repress gluconeogenesis. So we go with the first hypothesis, which is about the adaptive immune cells. So basically, now we start seeing mice. So basically, what we have here is we have two sets of mice. We have a normal mouse, which we call a wild-type mouse. So it has an intact B cell and T cell. So it means the adaptive immune response is normal. And then we have this mice that we call the RAC2 knockout mice. So these RAC2 knockout mice don't have T cells and they don't have B cells. So what that means is that upon any challenge that activates immune response, these mice will not be able to mount adaptive immune response. So in the context of the infection we care about, if we take these cells out, like these mice have, what happens to gluconeogenesis? So here again, we give a substrate of gluconeogenesis, this time glycerol, to the animals that are infected, the wild-type mice and the RAC2 knockout mice. So the wild-type mice are the ones in red, and the RAC2 knockout mice are the ones in black. So what we can appreciate here is that the RAC2 knockout mice, despite not being able to mount adaptive immune response, are able to produce more glucose from glycerol than the wild-type mouse that is also infected. So what this means is that if you take out the T cells and the B cells, and then you are having an increase in gluconeogenesis, what that means is that these B cells and these T cells are repressing gluconeogenesis. So we're still working on knowing whether it's the B cell or the T cell. And if it is the B or T cell, what subset of the B cell, T cells or B cells are really responsible for this? So, but going to the second hypothesis, which is about the parasite itself, we're keeping it simple. So in the question and answer, probably I'm going to talk more. So going to the parasite itself, in vivo. So what we did is infect wild-type mice. Now, we're not talking about RAC2 or any knockout. At a stage where you know that gluconeogenesis is repressed, you will then clear the parasite from a group of the mice that are infected. So basically what you want to see is that if you now take out the parasite, what happens to gluconeogenesis? And what we see here is that the ones in red are the mice that were infected and no parasite were cleared. So they still had parasites in the blood. But then the ones in orange, I hope I'm calling the right colors, but I'm using the cursor so you can follow. So the ones in orange, if truly it's orange, are the ones that we clear the parasite at some stage during the infection, and we check a few days after, because we want to ensure that the immune response is still intact, which we confirmed first that it is intact. So the only thing we're taking out are the parasites. And then you will see that the gluconeogenesis is improved because these mice now produce more glucose. So what this means is that both the parasites and the adaptive immune response are repressing gluconeogenesis in these mice, right? So what does this, how do we then know what's really happening? Because if we say that the immune response is involved in repressing gluconeogenesis, that can be intuitive. Sometimes it can be counterintuitive. But the fact that the parasites can do it is a bit intriguing. So we decided to just do a simple experiment to see what happens. So basically, is this, it's minimalistic because you can't really decipher this in a complex organism like the mouse. So we do cell cultures for this. Basically, what we do here is that we collect the hepatocytes. So hepatocytes are like the primary cells of the liver. We collect hepatocytes and put them in a medium, in a dish, basically. And then collect hepatocytes, put them in a dish together with parasites. So what you want to see here is to see the impact of the parasite on gluconeogenesis in the hepatocytes. So the simple readout, for instance, I've mentioned this enzyme, glucose-6-phosphatase, is to look at the expression of glucose-6-phosphatase. And what you will see is that when you have the hepatocytes with parasites, the expression of the glucose-6-phosphatase goes down. So we're still doing functional assays that shows that this is true. But for the purpose of this presentation, I'm not showing that, but I can answer it in the question and answer session. So it shows that the parasites can really do this. So, but independent of what the parasite does, independent of what the immune cells do, that the adaptive immune cells do, what is the implication of repressing gluconeogenesis for the disease outcome itself? And for you to understand this, you need to reverse it. So we're trying to rescue it. So if you have an animal that has gluconeogenesis repressed, if we stimulate gluconeogenesis, what happens to the animal in terms of the outcome of the disease itself? We tried different strategies, even giving the animals glucose as an example. With glucose, you're not able to maintain a consistent level of normal blood glucose. So what we finally resorted to was to use glycerol. So let's go through it together. The infected animal is the ones in red, and you will see as the infection progresses, the blood glucose level goes down. The ones in blue are also infected, but these ones receive glycerol for us to stimulate gluconeogenesis and increase the level of endogenously produced glucose. And you can see that the level of glucose goes up, although it's not as high as the initial level, but you have a significant increase in the level of glucose all through the infection. So what this meant for the outcome of the infection is that because the infection itself is lethal, so at some point the animal dies. If you look at a wild type animal, the ones in red that did not receive glycerol, on average, they die, let's say, on day 35, for instance, post-infection. But then when the animals are supplemented with glycerol and then they have a higher circulating blood glucose level, the survival of the animals are extended. And the normal thing that is common in parasitology, for instance, is that when you administer something to an infected animal and the survival is extended, if it's lethal, you often have a reduction in the level of the parasite, or sometimes maybe clearance of the parasite. That's to show you that, oh, the parasites are no longer there. This is why the animal is surviving. But in this context, the animal, the parasitemia, which is the parasite level in the blood of the animals, in the animals that are infected and did not receive glycerol in red, and those that received glycerol in blue are the same. This then takes me back to the first concept that I mentioned. I said what we often do, or what is common, is that you just kill the parasite. You eliminate the parasite. But what we see with enhancing gluconeogenesis is that the parasites are still there, but you've increased the capacity of the host, in this case the mice, to tolerate the parasites more. It does not mean that this strategy is better than this strategy, but this strategy can be a complementary strategy for this strategy itself. Because in some chronic infections, which is common in sepsis and some other chronic infections, sometimes you've induced so much damage to the organs of the host that even clearing the parasite will not rescue the human or animal, for instance. So this is a complementary strategy from the infection standpoint. But what is the bigger picture, which is where me being a physiologist, come into play. So, because this is not at the beginning of work on this, but we have a work ongoing in the lab at the moment where we've gotten 2 grants. So at least when you get grants for ideas, it means that someone thinks it's going to work or someone is taking a risk that hopefully it works. So we've worked extensively on how the parasite interact with adipocytes in the host. And we've gotten 2 grants, for instance, to see how we can then exploit this interaction to isolate compounds that can hopefully in future be used as like anti-obesity agent. It's still early stages. We have money to do this. This may not happen in 3, 4, 5 years, but that is the idea. So in the context of what I just explained to you, for instance, now in type 2 diabetes, one of the things that everything I explain is to make it very simplistic. But one of the things that happen in type 2 diabetes is that you have enhanced gluconeogenesis in the liver. And because the liver is producing a lot of glucose, it contributes to the hyperglycemia that you see in type 2 diabetic patients. But if I say that the parasites can repress gluconeogenesis, maybe in the future we can isolate compounds from the parasite that can repress gluconeogenesis in the context of type 2 diabetes. So we're working on this. Some things I cannot say because of the legal implication and patent implication, but just keeping it simple. But these are some of the ideas that we're exploring. Context of obesity, diabetes, and in the future, hopefully we're going to explore something regarding neurodegenerative disorders, for instance. So in summary, hopefully I didn't bore you, so I tried to keep it simple so everyone takes something away from this. What I've said in terms of the research is that in trypanosomiasis, for instance, African trypanosomiasis, you have a reduction in the capacity of the host to produce glucose. And then I said that both the adaptive immune response and the parasites themselves can repress gluconeogenesis, although experimentally we think the parasites do it better. And then if you can enhance the level of glycemia, not by giving glucose, because this is what happens in spurious hypoglycemia. These are very old reports of trypanosomiasis. So giving glucose doesn't really help because the glucose level cannot be sustained by just giving glucose, except you keep giving glucose continuously, kind of. So, and then the most important thing for me as a physiologist is that we're exploring this knowledge to find solutions to stubborn, non-communicable diseases. Obesity, my interest from the PhD, and then diabetes from some of the work that I did in the past. So this is to acknowledge my lab. So this is the head of our lab, and this is every member of the lab. So this is a PhD student in the lab. He's the one now actively working on obesity and trypanosomes. And yeah. And, these are every other person, I can't mention them, but these are the different nationalities in the lab. So these are the funding we have at the moment. We have funding from the government. We have one from the foundation and then from the institute itself. And I also appreciate University of Ilorin for letting me go do this research. Thank you so much. I think it should be the question session now. Let me minimize this. So thank you. So
thank you so much, Dr. Abdul Basit.
You're
This
welcome.
was a very insightful and educative session.
Thank you.
So let's check the chat box. Please, if you have any question in mind, drop in the chat box so that the doctor can address it now. I have I have some questions for you, doctor.
Okay.
It's it was very interesting anyways, and I kept hearing glucose, gluconeogenesis and all that. And the first thing that comes to mind is diabetes.
Yes.
And can we say maybe in the future or maybe currently there is a way that it can be related to diabetes, or is there a research ongoing for diabetes management?
So in terms of research ongoing for diabetes management, we are trying to do that. So I don't know if any other lab is doing that now, because the thing is this: I don't think what we do is super novel, but what we're doing is to bridge the gap between parasitology and physiology, for instance. So that's the innovation that we bring in here. So because the parasite, not just trypanosome, maybe other parasites do this, we can always find drugs that help in type 2 diabetes from them. Whether it will be better than what we have at the moment, that is debatable. You don't know until you get there. But the idea is that, you know, we want to spark the interest in people searching for solutions in unusual places, because we've been going to the usual. It's been the usual, for the parasitologist, we find something to kill the parasite. For the person working on diabetes, we actually still don't completely understand the etiology of type 2 diabetes, for instance. You have different factors, this and that, but maybe looking at this unexplored niche can help us find something better. Maybe we find something better, but if we find something, it's still a progress, in my opinion. So in diabetes, it's theoretically it can help. Practically things are more complex. So I'm not here to say that, I'm doing this and I'm finding the solution to that. No, science doesn't work like that, right? So, but we're making effort, and hopefully our effort or other people's effort culminates in making us healthier.
Okay, thank you so much. We're really hopeful that something great is going to come out of all the theories and practicals. Then
Hope so.
another question is based on the research on trypanosomiasis, is there any possibility or is there any discovery for maybe vaccine or drug or something that can actually help?
So in terms of the burden of trypanosomiasis itself, right? So in the context of human infections, it's not like malaria. It's not a disease that is killing people like malaria, as an example. So it's one of those, like, it's a bit neglected. Let me put it like that. So it is more a concern in cattle. You understand? So, and the vaccines, there's because we already have effective anti-trypanosomal drugs that are effective. So in terms of vaccine development, there's not so much going on for trypanosomiasis. There's not so much. For malaria, for sure, malaria is a big problem, right? It's a bigger problem, let me put it like that. So, but the thing, this is why sometimes, imagine if our focus of research is malaria, what that means is that the day we are able to eliminate malaria, there's no research to be done anymore, right? But imagine if we don't have malaria anymore, but then we can use Plasmodium to develop drugs for neurodegenerative diseases. It still makes the parasite important. So that's the same way the trypanosome is. So in terms of the human body, to a large extent, it's not a big problem. At least the ones that cause the human African trypanosomiasis, yes. There's some that are worse, but in terms of the burden in Africa, as an example, since this is a forum for Africa, so I don't think it's a big problem. So there's no vaccine effort for this
Effort for
at
now.
the moment that is active, no, that I know of. No.
Okay, thank you so much. Someone asked the question. The person said, can adding the glucose substrate to the host be the best solution?
Experimentally, so I don't understand what the person means by glucose substrate. If the person means the substrate for gluconeogenesis, or the person means glucose, so I'll answer it with the two assumptions. So first, if you supplement with glucose, it does nothing. It does nothing. We've done all these things. It does nothing. But then supplementing it with a substrate for gluconeogenesis that then leads to production of glucose improves the survival of the animal. So can you then use this as a solution? I mentioned in the presentation about the disease resistance and the disease tolerance concept. So disease tolerance will not replace disease resistance. But the advantage of targeting the tolerance and the host is that, imagine I have malaria and I have something that protects my organs from being messed up by the parasites. So it means that I'm healthy, but then I still need to clear the parasite. But imagine another scenario where having the parasite in me damages my organ, but I can then subsequently clear the parasites. So you will prefer the first solution of keeping the organ function intact, which is what I think the tolerance does, and then you clear the parasite. So it's not the best solution, but it's a complementary solution to a healthier living. Let me put it like that.
Thank you so much. I hope that helped. If you're still not in the clear, you can put it in the chat box. And someone said, are we likely to find same effects from other parasites different from trypanosome?
So let me tell you from the literature. So because this field is a bit unexplored. So in malaria, for instance, they've shown that the hypoglycemia can be explained because of the immune response, as an example. That happens in malaria. And then we are showing this in trypanosomes. I'm not sure if they've shown it in helminths as an I'm not sure they've shown it. I'm not sure they've shown it in helminths. So I will be economical in saying this. If it is a conserved response, it should happen with other parasites. But if it is not, you will see differences. I'm using viral and bacterial infection as an example, for instance. We know that in either of bacterial or viral infection, if you improve glucose level, the animal succumbs to that infection. And in another infection, if you decrease the blood glucose level, the animal is rescued. So it may not be a global phenomena kind of for all parasites, but at least for I'm a very economical person when it comes to responses. So you don't take it like everything. Scientifically, what we have data for, we don't know whether it's in all infections or it's specific to the ones we care about at the moment. Not so much report is out there.
Okay. Someone in animal health and animal husbandry said, how can this be cascaded to our farmers and people involved or interested in animal health and animal husbandry?
So this is me. Some things I will not say because of the legal implication. And when I say legal implication is that we're also in the process of trying to develop compounds for these diseases. So you don't want to let the cat out of the bag, because at the end, these things also have an economic implication, right? So for the animals, at the moment, I know that there are some more for the bacterial infections where they give glycerol, because they've shown that glycerol has like an antibiotic impact, kind of. So that works. But let's say trypanosomiasis in cattle, as an example, whether it will work or not, I don't know. But theoretically, again, talking about what we are trying to do in the lab. So what we're trying to do is we realize that if you can, let me not make it complex. So if you can restrict parasites to the blood without getting into the organs, you can actually make the animal live as though it has no infection. So we're trying to understand the factors and hopefully develop maybe drugs in the future that even if an animal is infected, it will prevent the damage of the organs. Theoretically, what I think this can help with is that imagine if an animal gets infected and it dies in 1 week, irrespective of whether you treat the animals or not, because it's an acute infection. So this strategy, what it can do for you is to prolong the survival of the animal, for the animal to respond well to the drugs that you give to the animals to target the pathogen. And I often use this in the context of COVID, for instance. The reason why older people died due to COVID was because their tolerance level to that infection is lower. So if we have, during the COVID thing, before the vaccines were developed, if we already had drugs that get the tolerance of humans, and it improved our tolerance to COVID, then it gives us time to develop vaccines to then give to those people. You get the point, right? So I will never say that this is the solution, but this is part of the solution. Thank you. I'm sorry if my response is a bit long. It's just to make it clear.
That's fine. We also have another one. Sometimes, someone says, sometimes I'm made to think that parasites, though harmful, could be seen as a path to treating some disease that are non-communicable. Could
Yes.
this be reasonable?
This is not only reasonable, this is practical, and this is something we are doing already. So let's think of parasite as a pathogen. So today we know what we do with viruses. When I say virus sounds like something you don't want to have, right? So, but viruses are being used to treat diseases. Not that the virus, imagine, not like you're being infected with HIV virus for you to get infected, but the mechanism that the virus uses to get into the cells have been used in diseases that actually have proved to have no cure all this while. Because you want to target, let's imagine you want to give a drug that goes into the retina, as an example. The best way you can target it is to have this drug together with a virus that is directed to specific proteins that are expressed by the retina. So the virus goes directly into the retina. The virus is not causing an infection, but what the virus used normally to cause an infection is now being used for the virus to do something positive. And this is something we are also exploring in the context of parasites, for instance. How the parasite infects you and make you sick can be explored to make you healthier in other contexts. And I believe we will get there. It's just a matter of time. We just need more people to bet on us to do this with more money.
With the last sentence, it is we need more funding for this.
Yes,
Personally, it will not go
yes,
through.
yes.
But based on what you just said, there's a question that relates to that. Someone said, I believe there are prospects in this research. Don't you think that extracting compounds from the parasite to repress gluconeogenesis could introduce the symptoms of trypanosomiasis in man?
So it is highly unlikely. So the reason why I say it is highly unlikely is that, so let me make it simple. So you're infected by a parasite. Your immune cells know that there's this stranger now. We need to attack it. So the compounds that will be secreted by the parasite to induce gluconeogenesis may not be an antigen that your immune cells need to produce, let's say, antibodies against. It is highly unlikely for this to be the case, because what we also know now is this. For the parasite, in this context, trypanosoma, to repress gluconeogenesis on the hepatocytes, they need to secrete something that goes to the hepatocyte. Basically, if you have a cocktail of secretions from the trypanosoma and you put it on top of the hepatocyte, that will repress gluconeogenesis. And this is me saying that I don't want to say some things yet, but we know that these factors are unlikely to induce activate immune response. Let me put it like that, because immune response is key to any development of sickness behavior. So it is highly unlikely. It is highly unlikely. Sorry, it's just for me to say that we use antibiotics, right? Antibiotics were extracted first from where? These are against bacteria, but these are compounds from pathogens also. So it's just like me saying that I take antibiotics and I'll be sick. So it's that sort of thing. So it's I don't want to say too much.
In this case, right, is it possible to translate this research into humans?
It's I know that you have so let me use diabetes as an example. If you go to PubMed and you type diabetes there as an example, you will see hundreds of thousands of papers where people did animal study, and they say that, oh, this leaf, this compound, that compound, this strategy can help in combating diabetes. I think, in my opinion, this is why I say I'm economical. It's a sales term. You want to sell, and then you say this. But we know that it takes a very long time. Whether this will later help in diabetes or not, if I'm writing it theoretically and I want you to give me some money, I will tell you the potential. You get my point? I'll tell you the potential, but I will never say that this is going to cure diabetes, because by rule of probability, it is more likely not to than true. So, but we're hoping that it does at some point. But I think this is not something that can happen in 5 years. So it takes time. The pipeline to now get to humans, it takes a long time actually, because our work on obesity, for instance, we've been doing it for, I think, 6 years. So, and it may not end in another 4 years. You understand? So, things don't work that fast, but we need to start, and we've started, and we've made significant progress. Let me put it like that.
Yeah, we need the funding fathers so that we
Yes.
can have something to build on.
Yeah.
Because we have young researchers that are looking at carrying out researches. There are so many challenges available. And you know that INSTAL Africa is all about capacity building and
Yes.
all the vision it has. If young researchers want to be involved in this in the studies
Yes.
now, how do they go about it?
So the first thing is, okay, you've learned something from here, and then you just go do your thing. That is one way, right? So, but
Yeah.
then if by definition of a young researcher is still someone who is a trainee, then it means you don't have the independence to start something on your own until you have independence. Or the other way is you join people that are already doing this. You probably have like master students. We have PhD students here. I'm assuming, right? So from listening to this may just spark the idea of like, okay, Dr. So person is looking at diabetes. Why not look at neurodegenerative disorders, right? Because it's novel. I think if you tell anyone that I want to use my knowledge of cerebral malaria to find cure for Alzheimer's, and you have preliminary data that shows that this can happen, a lot of people will take a bet on you. This is not you reinventing what others have done, but it's not like you're bringing the science from heaven also, right? So it's just being creative. I think people can leverage this knowledge, especially for people that are in like microbiology, who already have good knowledge of pathogens. They just need some knowledge of physiology, and you can use this as a basis for applying for maybe grants if you're already independent, or you use this as a basis for seeking PhD positions. The only caveat is that I don't know of 5 people worldwide that care about this thing. Care not care like because it's not important. It's just that people are not thinking of it this way. And it's just me being a physiologist, and I don't think I will ever be this is, how do you call it, imposter syndrome. So I don't think I will ever become a parasitologist, but merging both can help. But we don't really have people that are doing this at the moment. But you can work with a physiologist and you're a microbiologist, and you will bring the concept. Or you're a microbiologist, work with a physiologist and bring the concept. But if you're looking for a person who is already working on both, I don't think we have 5 people actually, because I know a lot of people in this field. So, but I don't think we have 5. So, because when I go to parasitologist conferences, I feel like an outsider because everyone wants to kill the parasite. When I go to physiology conferences, people want to solve diabetes, neurodegenerative disorders. They don't care about pathogens. So this is but it's an advantage, right? To some extent.
So you personally, what are the challenges you have actually encountered?
I will say that in terms of funding, I don't think there's a challenge because I'm privileged to be in a place and lab where we have enough money. I don't want to say we have a lot of money. So, but we have enough money to do, I think, to a large extent, whatever we want to do, right? So, but when I talk about the challenge itself, I think more of what I will do with this if I go back to Nigeria? So if you want to continue this research, and I think whatever I say that is a challenge in Africa is not news. So I won't go into all those. But what I was like, what INSTAL Africa is trying to do, what I think people should do is to improve their own capacity by having the right training. So when you have the right training, imagine in Rwanda as an example now, in the university, you have in a department, 90% of the people who have the expertise to do better research. I think we will demand for better. We were able to attract better grants because they are grants that are actually meant for Africa. And I told you my background. I went from neuroscience to metabolism and now to parasitology without taking a course in microbiology before. It's because I see an opportunity there, and I know that in Africa, one of the things you will be funded for is infectious disease. And then I'm working with a lab that is doing molecular parasitology. And then I bring my knowledge of physiology to do this thing. I learn parasitology and I bring physiology to the table. I don't think I have any challenge here. But in the context of home, we have a lot of challenges. Yeah, which will not be news even if I start mentioning them.
Yeah, so several bottlenecks. Political bottlenecks will be the number
There's
one.
politics everywhere. There's politics everywhere. There's politics everywhere.
Wow. Someone says she wants to be involved. Is there like an opening in your lab, or is there a period where they bring in people?
So my lab is Gulbenkian Institute of Molecular Medicine. So that you've seen from the presentation, you can check. So openings will be there. In my lab, we don't have an opening officially. But imagine if the person wants to come as a postdoc, as an example, which means the person has some experience. You can always write to the lab. And sometimes positions are created because a person who we found very worthy, and we don't want to, I think it's just like football. You want to have the best players, right, in your club. So sometimes because there's an opportunity, you go for a person, not because you really want to do it at that moment. That is one way. But then we also have PhD program. We have a PhD program here that people can apply to, so long as you show your competence, and it's very international. So you want to have people from different parts of the world. That is another way. It doesn't guarantee that you will work on my lab, but it guarantees that if you're admitted, you will work in an environment that will improve you a lot. You understand? So that's another way. So the other way you can get involved is if I manage to find funding in Nigeria, you want to come to Nigeria to do research. But that we will also do in the context of neurodegenerative diseases. And we're looking for money to go back home. So hopefully we get money.
And I'll pray for you. Amen. Thank you so much, Dr. Amin, for this valuable insight, the questions, the answer. Thank you so much, everyone. No more questions here. In case you remember any question and you want to ask, you can actually drop it, and then I will get across to you after the event. I thank everyone for taking their time to join. So thank you so much, Dr. Amin, for taking your time to show this wonderful presentation.
Thank you so much. You're putting together something very fantastic, and I must applaud the professionalism of the team. Even when I saw your website, I was like, wow, it's my little knowledge of web design, like, oh, this is done very well, So I keep it up, and I hope that we get better with time also.
Yeah, we're getting better. We're getting there gradually. So thank you so much, everyone, for joining us today. Please don't forget to share this with your friends, families, on your social media. If you haven't registered, please register so that you can receive our mails and know when we have other events. And we usually host every month different events. We have from the nurses to the maternal health, we have to research methods, qualitative data analysis, and I'm sure you enjoyed today, so you will not want to miss the other conversations that are coming. And thank you so much as we build a thriving community for research. We pray that we get more fundings, we get more people that want to be involved, and we'll be able to meet up our project and our goal for the INSTAL Africa. So together we can make a wonderful team and make wonderful research group, research community, research country. Thank you so much and have a wonderful day to
Goodbye.
everybody. Okay, bye.